Our project was on the structural analysis of the phenotype of the
generally the animals get more and more irritable.
proline 191 mutant heterotrimeric complex. So basically, the heterotrimeric complex that we're working with PTF1 is it's a master regulator of pancreas development, and what prompted this study was we found our mentor in a previous experiment, or in a case study, he found a proline 191 to threonine mutant in a Turkish family, and so what happened as a result of that was their pancreases did not develop. They had pancreatic aplasia, and so just a brief overview of the actual complex itself. Which is over here. You have the E12, PTF1A, and RBBL, and then there's another variant, the E12, PTF1A, and RBBJ. So there are two, because one is responsible for the early development of the pancreas, and then this RBBL is responsible for the late development of the pancreas. Yeah, so the major difference between them is that the bonding between the PTF1A and the RBBJ includes both a bond between RBBJ and the PTF1A at the C1 and C2 positions, while the bonding between the RBBL and the PTF1A only includes it at the C1 position, not at the C2 position. And so that's like the major structural difference between the two. All right, so in early development, what happens is that they basically the structural cells, the acinar cells of the pancreas are developed, and then in later development, what happens is that the pancreas further develops, and then there's a protection of digestive enzymes for the function of the pancreas. So in that previous study, the case study, what they found was that due to this proline 191 to threonine mutation, which occurs, if you were to look at the figure, this is the same thing as what was over here with the RBPL. Here you have your RBPL, you have your PTF1A, you have your E12. And so at this position right here is where the mutation occurs at the proline 191, which is right here. Due to this proline changing to a threonine, what can be seen is a 75% decrease of forming the PTF1A E12 heterodimer, and a 66% decrease of the transcription function of the PTF1 heterodimer, which basically means that it was losing its interaction with the DNA, and as a result, not as much mRNA was forming, which would go on to produce proteins that would develop the protein, and the acinar cells needed to develop the proteins. So our question was, or what we were trying to do was, we were trying to predict basically the structural changes that occurred due to that mutation, and like how does it look? How does the interaction between this heterotrimeric complex change as a result of the mutation? So in the wild type, which is the original before the mutation, at this proline 191, what you'll notice is that there's this basic loop helix structure where you have a helix where it coils, and then there's a loop, and then another helix where it coils again. So the proline 191 is responsible for telling the protein when to stop coiling and then begin its loop into the next helix.
Yes.
So when this mutation occurs, what happens is that in the mutant, there's an extra helix, because it wasn't told by that proline 191 due to that mutation that there was that to stop coiling, and as a result, the loop shortens up because there's an extra. As you can see, there it goes. There's 1, 2, 3 helical turns, and in this one, there's 1, 2, 3, 4 helical turns. So at this point right here, roughly, there's a threonine 191 at the position 191, in contrast with the proline here. And so because of this, if you notice in the original, in the wild type, you have an arginine 198 and a lysine 201 interacting with the DNA. So due to that interaction is what causes production of acid in our cells. It basically increases the interaction. So when this occurs and then the loop is shortened up, what happens is that this arginine 198 and lysine, they stick out and away from the DNA, which decreases the interaction with the DNA. And causes it to basically reduce the amount of acid cells that are developed, and then basically cut off the development of the pancreas.
Okay, so wonderful explanation, thank you. So just as a review for me, who really appreciate this, point again to what is the normal non-mutant figure.
Right.
So this would be the wild type, which is what you call the normal.
Okay.
This is what you see normally in nature.
Okay.
So as you can see, at this specific point is the proline 191. That's where normally it's going to be at this position, and normally it stops the helical turns at this point, and then it allows the protein to transition into a loop, and then back later on into a helix, which is like the coiling.
Yes, and someone with the mutant development.
Yeah.
Tell me again what sort of issues, health issues and such, they would have, and how prevalent is that running in families, or I know you mentioned certain people from certain parts of the world.
Okay, so due to this mutation, what happens is that there's less interaction with the DNA. And so since this is a master regulator of the pancreas development, the problem that results because of this is that
Viewing to stop
babies are
sentence.
pancreas,
Oh my
and therefore they do not have the insulin they need to survive.
Interject some.
So this could prevent the baby from even being born full term. And so in studying this, if you have identified that perhaps Kristen has this mutation in her family, do we then go and test other people in Kristen's family? Do we try to identify potential health issues that could possibly be corrected? Have we gone that far in our research with that?
Well, for our research,